Hello there everyone, my name is Anthony de Vinagracia and today I will be going over with you a lecture on amino acids. The following slide here is just a brief explanation on the learning objectives of what we hope to accomplish during this presentation. So let us begin, shall we? Now, amino acids, to give a brief definition, they are technically chiral organic compounds that contain an amino group and an acidic group. There are many thousands that can be made. However, there are only 20 different amino acids that are used to make up proteins. Speaking of proteins, one of the main functions and purposes of amino acids is that they are used to build up proteins. However, though, they do have several other functions, such as the production of neurotransmitters like serotonin and norepinephrine by the amino acids tryptophan and tyrosine respectively. They are also very helpful in maintaining cardiovascular health, as the amino acid arginine is used to make nitric oxide, which is helpful in lowering blood pressure. Overall, amino acids, not only do they have the capacity to polymerize, but they also have varied physical properties and chemical functionalities. Over here we have just a brief picture of the anatomy of what an amino acid looks like. As you can see here, they contain an amine group, an acid group, centered with an alpha carbon, and the most important part at the end, an R variable side chain. This R variable side chain, or R group, is a very important aspect, as its most important part in the amino acid is its polarity. When it comes to amino acids, out of the 20 that I mentioned earlier, they can be classified based on the R group and are divided up into 4 groups: nonpolar, polar neutral, acidic, and basic. The link that you see here below is a video that goes further into detail on how these amino acids are divided out into these 4 groups. You can take a look at the video when you have the time after this lecture. Right over here, we do see the 20 common amino acids divided up into the 4 respective groups as I mentioned earlier. Out of all the 20 amino acids, they all fit the same formula. However, one of them, which is proline, doesn't exactly fit the same formula as I shown in the previous slide, but it comes pretty close enough. Next thing I would like to talk about is how to differentiate between a peptide and a protein. Now, peptides and proteins, they are similar in which they are both made out of amino acids. Both of them are formed by amino acids that are linked together in peptide bonds. One major difference is when it comes to a peptide only contains 2 to several dozen amino acids. Examples of peptides include oxytocin and vasopressin. However, it would be considered a protein if instead of a few dozen, there are more than 50 amino acids that are connected together in a chain. An example of this is the hormone insulin, which is a glucose regulatory hormone that helps control your blood sugar in case it gets incredibly high. Now then, moving on to one of the major topics in this presentation, amino acid metabolism. A lot of you are familiar with the fact that if you want to gain mass or gain energy, you need to eat more protein, so to speak. Well, in a sense, that is kind of true. Now let us dive into the actual scientific facts about that. Out of the 20 amino acids that I previously shown before, humans can only synthesize only 10 of them. Examples of these 10 include valine, proline, and tryptophan. These 10 essential amino acids, they are obtained through the consumption of proteins, vitamins, and supplements. When these amino acids through these proteins are digested, they are broken down and are used to provide to the total daily body's energy requirements. The video link that I showed here below is a pretty good video that does a good, pretty more detailed explanation on the essential breakdown of these 10 essential amino acids and the cycles that they go through once ingested in the body. But for now, I'll give you just a brief, good general outlook onto what happens when they are ingested. Overall, over here, we have just a brief outlook into how amino acid metabolism works out. Basically, once these proteins are ingested and broken down in the digestive system, the amino acids are absorbed into the bloodstream and sent directly to the liver. Once in the liver, they are organized and arranged into amino acid pools, so to speak, and they are distributed to different tissues and areas of the body that are in need of them for energy and protein buildup. One example of this is some of these amino acids, they are immediately converted into intermediates of the citric acid cycle, which is one major important cycle used in the production of ATP or energy for the body. Any amino acids that end up being produced in excess would be sent through the urea cycle, which would be sent to be excreted out of the body through the urine. Next thing I would like to talk about are aminoacidopathies, which is the name given to basically inherited disorders in amino acid metabolism. There are many types of these aminoacidopathies, but for today, I would like to go over with you 3 of the most common types: phenylketonuria, maple syrup urine disease, and cystinuria. For phenylketonuria, it is basically an inborn error that is caused by the absence of the enzyme phenylalanine hydroxylase, or PAH, which its major responsibility is to help in the conversion of phenylalanine to tyrosine. Due to this absence, the amounts of phenylalanine end up increasing, and this high levels can lead to seizures and behavioral problems and mental disorders. The treatment options for this disorder include starting a diet on food that are low in phenylalanine. Keep in mind, though, that this diet, once a prognosis is made that the person has phenylketonuria, the diet should be started immediately and it would last for the rest of their life. Over here is just a brief picture that pretty much is a visual representation on the results of the absence of phenylalanine hydroxylase in PKU. Examples of foods that are low in phenylalanine are your vegetables, fruits, juices, and low protein breads and pastas. Once again, keep in mind that the patient with this disorder would be going on this diet for the rest of their life. There is a screening test for phenylketonuria known as the Guthrie test. It is basically a bacterial inhibitory assay that uses the properties of phenylalanine to induce bacterial growth on a culture that has an inhibitor. A drop on the infant's blood is placed on a paper disc, which is then placed on the media. After incubating the media, if there is bacterial growth, then the patient is positive for PKU due to the high amount of phenylalanine within that drop, which help the bacteria overcome the inhibitor. Next up over here is maple syrup urine disease, or MSUD. Basically, this disease over here involves a defect in the inability to get rid of leucine to convert leucine, isoleucine, and valine, which leads to increased buildup. Clinical features of this disease include mental retardation and can lead to death if not treated soon. The reason it gets the nickname maple syrup urine disease is due to a maple syrup-like odor within the urine. Like PKU, the screening test for this disease is also a Guthrie test. The only difference is that the inhibitor would be 4-azoleucine. The treatment options for MSUD is like before a protein-free diet, or basically any foods that are low in leucine, isoleucine, and valine. Right over here is an example of a diet powder that an infant would have to take that does not contain any of the three mentioned amino acids. For cystinuria, the next one, it is basically an inherited recessive disorder that is caused by a defect in the amino acid cystine's transport system. This leads to an accumulation of cystine stones in the kidneys, ureters, and bladder. A urine screen test for cystine can be done by using cyanide nitroprusside. If the urine turns red-purple in color, the patient is positive for cystinuria. Phenylalanine within the drop helps the bacteria overcome the inhibitor. Some treatment options for this disease is to just start the patient with a high fluid intake in order to prevent the formations of the stones. If this does not work, then the drug penicillamine is prescribed, which works as a chitling agent, which its main purpose is to break down the stones. So overall, as a summary, today I was able to go over with you several of the more important functions that amino acids do to the body. I was able to differentiate with you between an amino acid peptide from a primary protein. I also gave you a brief explanation on amino acid metabolism, and also went over to use several aminoacidopathies, most of them in which involve a defect or the absence of a certain enzyme which prevents the conversion of an amino acid to another form. And that concludes this presentation. I thank you all for taking the time and listening to this presentation. I hope you all learned something from this. Once again, like before, if you want further information on amino acid metabolism or the way that they are classified, please check the video links that I had on the presentation, and also take the time to take a look at my other learning activities, 2 case studies related to several diseases, to several aminoacidopathies, and test yourself to see what you have learned. Also, make sure to take a look and take the time to fill out the instruction questionnaire to see what did you think about this presentation, what did you enjoy about it, what could be improved from it, so that improvements can be made for more better presentations. Thank you all for your time, and have a wonderful day.